It's a pleasure. It's a pleasure to be here today. You heard a little bit about my background. I started to be interested in genetics as an undergrad student when I was studying at McGill University, and being from Quebec, you may know now that we are a population that is maybe a little bit more aware of genetic issues, since we're a founder population, at least parts of French Canada or Quebec. So after my studies, my grad studies, which were mostly focused on genes controlling cardiovascular disease, when I arrived in Puerto Rico, I noticed that there was very few resources available for patients in terms of hereditary cancers. And I had experience already with the BRCA genes, so my first project was really to look at what is the characteristics of hereditary cancer in Puerto Rico, and how can we help, or how can we work to try to give better access to patients here to what is now standard of care in genetics in the US. So what I'm going to be talking about today, I'm going to give you an overview of the work that we've done in Ponce about genetics of breast cancer. And the talk is going to be divided into hereditary breast cancer, but I'm also going to be talking about ancestry and its role in the cancer risk in Puerto Rico. And if I have time, I'm going to start covering some of the studies looking at the genetics of more common or complex breast cancers using SNPs and copy number variants. So it's going to be kind of a nice follow-up to what you have so far, as these are going to be a mix of clinical data, but also research data on what is the current status of breast cancer genetic in Puerto Rico. So when we talk about Latin America, it's a very wide term that actually designates different populations that really have not so much in common, maybe apart from language, but that exclude Brazil, some cultural aspects as well. But in reality, what I hope I will convince you of today is that they're actually genetically different populations. And so when we look, and why are we really worried about Hispanic and Latino population, is that they're a very important part of the US. And you can look in blue are the areas or the counties where you have more than 50% of Latino or Hispanic, most of them being probably Mexican Americans, Cubans, and Puerto Ricans. And so there are 8.5 million Puerto Ricans, and if you're studying here, or if you're going to be practicing here, probably you are one of them, or you're going to be directly working with this population. And they make up 18% of the Hispanic population in the US. So it's a small island, but it's a lot of individuals to care for. So you should know by now very well that first-degree relatives of women with breast cancer are more likely to develop the disease than the general population. And that part of this risk, at least, is due to inherited predisposition. So we know that, and you've seen this many, many times now. So we know that hereditary breast and ovarian cancer is a small proportion of all breast cancers. And the rest are those complex breast cancer that can have more or less environmental contribution. And so we're going to talk a little bit about the hypernetrans susceptibility genes, which traditionally were BRCA1, BRCA2. There's more of those genes that are known, some of it with higher penetrance and some of it with lower penetrance. And I think that eventually they're going to make their way into the clinic. But so far, BRCA1 and BRCA2 are the ones that are most commonly tested in the clinic. And there's also all of those in the last part of this graph, where we have many low penetrance, so genetic factor that will have a little bit of impact on the risk, but it's going to be working together with the environment, and it's going to be a combination of small risk factors that are going to make women or a given family breast cancer risk. And we're going to be covering the BRCA genes, and I'm going to be talking also about some of those low penetrance susceptibility genes. So what is the status of the BRCA mutation in Hispanic and Latinos, and how does it compare? What is the status of genetic testing for the BRCA genes in Puerto Rico? This is the first things I'm going to be talking about. And I'm going to be presenting the data from a study that was done in San Juan and Ponce, where we basically have looked at women that have had a BRCA test done with clinically with Myriad Genetics. And we have looked at what mutations were identified, what were the family characteristics of those populations, and whether the tools that we use in the US to predict the risk of a person being a carrier of the mutation were actually valid, or how did they perform in the population here. So it's a retrospective study, and it's an ongoing study. So the sample size is still relatively small, but we're getting to start an idea of the picture. We also asked the patients about insurance coverage. So we wanted to see how many people had to pay out of their pocket the BRCA test, and how many people actually had an insurance coverage. And this study is from 2 years back. So this is still evolving, but I'm going to be able to give you some information, because the status of insurance coverage of BRCA testing is unfortunately not as good as what it is in the US, and this is something that we need to keep working on. So this is the family history of all of our cases. So you can see that we don't have a pointer, right? So you can see that we have a total of 23 cases. 2 of those were BRCA1 mutation carrier. We had 9 BRCA2 carriers, and we have 12 non-BRCA carriers. So those are the people that have had the BRCA test, and the test came back as being negative, no BRCA mutation. And you can see that a large proportion of our positive cases had an earlier age of diagnosis. And we have gathered all the family history of cancers for these patients. So you can say that some of them had ovarian cancer family history, and we also saw a few other cancers in those families. Now, what we did with those data is we put it through carrier prediction risk models. And so I don't know if you have heard about the Myriad carrier predictor model, which is actually on their website. It's a site where you just go and you're going to enter a patient's information, and based on all of the data that Myriad has gathered since they have started clinically tested BRCA testing, they're going to tell you a percentage of risk that that particular person is a carrier of the mutation. So this is purely based on empirical data. It's based on all of the data that is available from previous Myriad tests. So what it's asking about is asking about basically the sex of the person that you're seeing, whether it's an Ashkenazi Jew descent individual. Do you know why they ask about this? Yeah, because it's a founder population, but they have a higher risk of BRCA carrier for the same family history than other populations. So that's something they ask about. They ask about whether the patient has been diagnosed with breast cancer, what was the age of diagnosis, and they ask about the family history of breast and ovarian cancer. And once you enter all of that information, it computes a risk for that given person that that person is a carrier. And some practitioners will use that together with other tools to decide whether the person should be offered genetic testing or not. So it's a very important tool to be able to know whether you're going to be offering a test to someone or not. So it's important that these models are accurate. But as you can see, they don't ask whether the person is Hispanic or African American. So the only data, population that they're specifically interested is Ashkenazi Jew. Now there's another model that allows you to do something similar, but that takes a little bit more information into account, and that's the BRCA Pro model, in which you're going to enter the actual pedigree, and it's going to compute a risk based on how many relatives have the disease, their ages of diagnosis, and so on and so forth. And so you can see that the BRCA Pro model is going to give you a risk estimate for BRCA1 and BRCA2. And if I enter information similar to the one that I had in the table before here, I had a risk of 10.4% for that women. Now in the BRCA Pro, you can see that the Myriad risk is still 10.4%, but then the BRCA Pro will give you a slightly higher estimate. Now we did this, we put the data from our patients and family through the Myriad and the BRCA Pro model, and this is what we obtained. So what you can see on this graph is the carrier risk estimated by BRCA Pro and then by Myriad. And you can see that Myriad always estimate a little bit lower the risk than BRCA Pro, and this is normal. And then you can see that the BRCA negative cases are more towards the lower end of the graph, which means that they were identified as being a lower carrier risk of BRCA. And then the BRCA2 carriers are in the middle, and the BRCA1 carriers tend to have a higher risk estimate. And the reason for that is that if we come back to our families, the ages of onset of our BRCA1 carrier was much higher than the ages of onset of our BRCA2 carriers. And this is when we were talking about differences in BRCA1 and BRCA2, this could be one of them. However, we can see that 2 out of our 9 BRCA2 carriers obtain a risk estimate of less than 10% for both models. So they were BRCA carrier, but the model gave a very low risk estimate. And this is where it becomes really important to actually look at the family tree and really think about whether you should be offering testing to those individuals or not. However, since they both had an age of onset that was very low, below 45 if I remember well, then they would meet the guideline for the NCCN guideline for testing. So those individuals would probably have been offered testing. There was one individual that obtained a risk estimate of less than 5% for both models that wouldn't meet the NCCN guidelines, but that tested positive. And so that person is one of those persons that is carrier of the mutation and would have gone untested. And this is something that happens. We want this to happen the least possible, but there's some situation where really we cannot tell from the family history that a person is a carrier. So are those, and we're currently gathering more data on this, but we can ask the question whether those models that are not really specifically designed for different populations would work equally well in Hispanic or African American or white population in the US. And some papers have looked at this, and what you see here, and the results have been mixed. And so in this paper here, what they found is that the BRCA Pro models were performing equally well when they were comparing Hispanic and other populations in the US. But there was another paper that was saying that the models were performing much less in Hispanic population. And the reason for this discrepancy is that, well, maybe they were looking at different Hispanic population, so we don't know. So that's why it's very important to treat or to look at each population specifically and do those assessments for each different population, and not assume that by doing this in one Hispanic population from California, which is most likely Mexican American, it's going to apply to all of the Hispanic population in the US. And the importance of doing that exercise is that some studies suggest that the prevalence of the BRCA mutation would be a little bit higher in Hispanic. That has not been verified here yet. But this is comparing the risk carrier estimates for individuals that would be of non-Ashkenazi Jew ancestry and those that were of Ashkenazi ancestry. And this is part of the myriad table. So you can see that for a person that doesn't have that didn't have any breast and ovarian cancer, but that had and that has no family history, the risk is 1.5%. So this is kind of the general population risk. So if you take 100% in the street and you do a BRCA test, you expect to find 1 to 2% with BRCA mutation. However, if you do the same exercise with Ashkenazi Jew ancestry individual, then for the same exact family history or personal history, that risk increased to 8.2%. So it's very important to get an accurate estimate of the risk. It's very important to have that data for each population that you're going to be working with. Now, if we look at what are the mutations that we found here in Puerto Rico, we don't know yet if it's a founder population. There's some indications for other genetic disease that it might be. But what we see is that we have a few mutations, especially the BRCA1 deletion of exon 1 and 2, and then the E1308X, which are mutations that are seen in many patients, despite the fact that we have a small sample size. So those are mutations that we think might be maybe more prevalent in the population. Also, and so how does this compare to other studies? Here I'm comparing our data with data that was done on Hispanics from California, so mostly Mexican American. We found in Puerto Rico that the BRCA2 mutation so far was much more prevalent than the BRCA1 mutation, which is kind of opposite to what has been seen in Mexican Americans. And now if we think that ages of diagnosis are a little bit later with the BRCA2 mutation, then maybe it's harder to see those families as high-risk families here in Puerto Rico. Now if we compare which mutations we found, we have our 3 more prevalent mutations here in Puerto Rico, which account for most of our cases so far. If we look in Mexican Americans, we don't find any of the mutations that are very prevalent here in the Mexican Americans from California. So the mutation spectrum in both populations is completely different so far. Their most common mutation is the 185 del AG, which is an Ashkenazi Jew, one of the 3 mutations that is very prevalent in Ashkenazi Jews. And this mutation we don't find at all in Puerto Rico so far. And I can tell you that we have more BRCA positive family, and I still have to see that 185 del AG mutation. So mutation spectrum is also very different between the both populations. So what we have so far is 6 different mutations, but 3 of them explaining 3/4 of the case. And we have drastic differences in the mutation spectrum and BRCA prevalence among different Hispanic or Latino population. And we saw that the BRCA1 mutation carrier had a higher score for both Myriad and BRCA Pro with risk computation. So there's less chances that these patients are actually missed and that are actually not offered a genetic test that they would need. So we were lucky to have those patients that got BRCA testing, and actually I can tell you that those patients were recruited in hospitals in San Juan and in Ponce, mostly in San Juan, that are known to be higher socioeconomic status hospitals. So, you know, it was more likely that these individuals will have insurance coverage for the BRCA test. So in the ideal world, and you heard about genetic counseling before, a patient will have a genetic counseling session where they would be educated about their risk, have a risk assessment, and then would be offered genetic testing, and then the person would be seeing a genetic specialist again after receiving the results. And there's probably more than one visit so that you can have a chance to offer the education and the testing to many family members. Well, from my experience in Puerto Rico so far, pretty much none of this is done. So the genetic test is often done in the surgeon or oncologist office, just as any other test that would be prescribed in the office. So, you know, you're going to order a pathology for receptors of the tumor, and then at the same time a BRCA test is ordered. So many times the patient are in front of a result of a test that they didn't know what it was, and so the education is kind of done afterwards, and if at all. So that also means that for those families that have a BRCA negative test result, but that are still familial cases or that are still at a high risk of breast cancer, they may not get that very important information that they need to keep doing the screening with a little bit more frequency than the general population. We also looked at how much money people spent on their test. And we can see that most people that actually got the test had insurance coverage. And so they were paying, at that time it was $3,100, but this keeps going up. But they were paying about 10% for the deductible, sorry, for their test. Now, there's some individuals that actually didn't have any insurance coverage and went ahead and paid for the whole test. And if we looked at all of the tests that have been ordered in the clinic, we found out that more than 50% were canceled because the patients didn't have insurance coverage and didn't have the money to pay for a test. Especially if they're in cancer treatment, they have a lot of expenses. Now things are improving a little bit. The 3 of the main medical plans are covering sometimes the test, but still it's very difficult to have relatives covered, especially if they haven't had a cancer. It's very difficult to have individuals that didn't have a cancer but that are at very high risk covered. So there's still a lot of education to be done with the insurance company, and even an insurance company that is the same company in Puerto Rico and in the US, they have different policies for both territories. So what would be helpful also is to see that, well, if we can confirm that we have only a subset of mutations in Puerto Rico, then we could think of a panel that could be done a little bit like the Ashkenazi-Jew panels, where a smaller number of mutations would be screened prior to do a full-blown BRCA sequence analysis, which is the one that is so expensive. And actually, Dr. Weitzel at City of Hope in California has developed such a test in Hispanic. And basically what he's doing is gathering all data from Hispanic population and trying to devise one of those lower cost tests that could be performed as a first screening in the hope of making the test more widely available to women. Now, what do women think about this? You know, we talk about the science, we talk about the risk calculation, but we often forget about what is the process for women and what do they know about it before getting their test, and do they want to hear about it, and do they want to be educated about it. So I don't know if you had a chance to hear about this, and I don't know how my internet connection is going to be, but if you want to have really an idea of what is the process when a woman have a family history, gets the test done, and what kind of thought process she goes through, there's a very nice movie that was made a few years ago. It's called In the Family, and it was, I think, made by PBS, and I might be able to show you the trailer.
Medical researchers have isolated a gene linked to a type of breast cancer that runs in families.
You know the numbers. You've got about an 85 to 90% lifetime risk of developing breast cancer, and about a 50 to 60% chance of developing ovarian cancer.
I think about my breasts and my ovaries as, when are they going to go off? Are they going to cooperate?
I started thinking about losing you.
You're not going to lose me.
I'm like, she has a 95% lifetime risk of developing breast cancer unless she has her breast removed. Does she only like me because she wants my babies? And she wants them quick?
My grandmother was only 38 when she passed away, so I never met her. And my mom always waited till that birthday. She kept thinking, this could be my year. So it's always there. She did get it at 50, died at 54. I decided that I'm not going to wait around.
A controversial new television ad is making headlines this morning. It urges women to get a genetic test to see if they are likely to get breast cancer.
Breast cancer runs in my family. My mother,
My grandmother?
my dad's sisters.
Myriad's charging $3,000 to do one test. I hope it doesn't turn out the case where it's only available to those women who have the resources to pay for it.
I myself decided to go and take the genetic test.
I think that most black people don't go seeking stuff because we got enough stuff to deal with already. Right. So we don't have the time to go seeking out more stuff.
See how my cancer stressed people out, they can't even deal with me.
Yeah, but you know. I have no breasts, I have no hair, I have no ovaries, I have no uterus. To say that, oh, it doesn't matter is a lie. It sucks. But being alive is what matters. And in retrospect, if I could have turned the clock back, I would have had all those surgeries.
I'm just moving closer to feeling that. The breasts and ovaries, you can live without them. I know I can't. It's just, I'll be back.
So that was just a quick, oops. way for you. So if ever you're interested in, seeing more about what kind of process women go through when they face that decision of not only doing the test, but after the test is done, whether they're going to get preventive surgery or whether they're going to be, keeping their breasts and having more frequent mammography, this is, I think, a very good perspective on that. And I think you can find it on the PBS website. You can order the movie. So we went into the population here, and we went to see women that actually were at risk from having some relatives with family history of breast cancer before 50. And that was a collaboration with behavioral scientist Dr. Varapalampil in Moffitt. And so we were asking them about what they knew about the genetics of cancer and what were their interests, in which occasions would they get tested, or what was important for them to be tested. And we did the same comparison in women that were from the Tampa area in Florida to see if there were any differences in what women would think if they were Puerto Rican from Puerto Rico or from the US, since the environment is a little bit different. So basically, what women told us is that they were very, very preoccupied with making choice about their children's health care. And that was the number one motivation for them to decide to do a genetic test. So it was more for their children than for themselves that they were interested in knowing the test results. And that was really the number one reason to do genetic testing for breast cancer. And then in second came only if results would help to make choices about healthcare. And we found out that one of the very important facilitator to get genetic tests done was the recommendation of their healthcare provider, and that is being their physician. And we know that genetic testing should be non-directive, so you should give the information to the women, but in the end, it's for themselves to make the decision. But what we found out with women here is that they're often not really comfortable about making a decision themselves about doing a genetic test or not, and they really like to hear a recommendation for their physician. That also means that if we want to make the test more available, it really goes through getting the physicians to talk about it to their patients, which might not be done enough right now. So what would prevent women from undergoing genetic testing? The cost was one of the most important factors, and women are right in the fact that many times insurance doesn't pay here, and as insurance plans starts covering more and more the cost of the test, we need to pass that information along to the women so that they don't decide not to make a genetic test just because they think it's going to be very expensive. Also, some of them, which was interesting, were worried that the test would be too painful. And this is really an indication that there's maybe a misunderstanding of what a genetic test is, as now the Myriad test can be done with a saliva sample, which is not what most people consider painful. So there's really a need here for educating women about that genetic test. So we found out also that over 92% of the women were unaware of the prevalence of the breast cancer gene, and 100% of the respondents agreed that they would seek genetic counseling in the next 6 months after the study if it were available. And the problem is that we know that in Puerto Rico it's really not very widely available, and especially if you're thinking of adults genetic testing. Most of the genetic counseling, is done for pediatric cases. And the most commonly cited barrier was associated with cost, which is something that is probably going to change in the future. So we've have an idea of the situation of the BRCA mutation in Puerto Rico and BRCA testing. And now what I want to move on to is the role of ancestry in breast cancer risk and in tumor presentation. Did you have a little bit of a talk about ancestry in genetic of cancer so far, or we're starting from? Okay. So, well, what we call, and so Latin populations are what we call admixed population. And what does admixture means? It means the act of mixing or mingling. And the mix that have occurred in most of the Latin America country is when the people from Spain arrived and they colonized the territory where the Native American were living, and they brought with them those slaves to do all of the hard work because they were not really into working hard, right? So in genetics, when we refer to an admixed population, we refer to the intermixture of previously isolated population. And we're talking in this case about the Spanish population, the African population, and the Native American population. So how do we study the ancestry of those populations? We need to have a way in our genomes to be able to say what part of the genome of a person is coming from which ancestral population. So we have what we call ancestry informative markers. And you may be aware of what a SNP is, a single nucleotide polymorphism. It's a one base pair change in a particular site in your genome. Well, the AIMs, or ancestry informative markers, are just like SNPs, except that they have a specific characteristic. We find that between the populations that we're interested in, for example, if you want to compare European ancestry and African ancestry, you're going to find a high differences in the frequency of the alleles of that SNPs in the different populations. So if you go into someone's genome, and let's say that this allele here could be an A, for example, and the second allele would be a G. You go in the genome of a person and you find out that that person has an A allele, which is the one that is very frequent in population 1, then most likely that part of the genome is coming from population 1. So you can do statistics on that by looking at the genotype of those ancestry informative markers. You can tell from which population is coming which part of the genome. And you can do that with a number of those AIMS markers throughout the genome completely. And that what that gives you is that gives you a proportion of the genome that is coming from each ancestry that you're interested in. So this is how this would look. This is actually data from Puerto Rico. And so we're looking at the genome of about 500 individuals, and each line on this graph is a different person. And there's 500 of them that are plotted on the same graph. So the pink part is the proportion of the genome that is from African ancestry. Now the green part is the European ancestry, and then this part here is the Native American ancestry. So what that means is that you have some of those people that have a very strong European component, but then almost no African component. So these are people that didn't inherit any of the African ancestry in their genome. And then you have people that are almost completely with an African background and very few European parts to their genome. And this is characteristic of the population of Puerto Rico, is that there's a lot of variability along the proportion of ancestry that is from European versus African. And in terms of the Native American ancestry, it's a small proportion, about 11%, and it's pretty constant throughout the population. And this is unlike other populations that are in Latin America. So if you look at the ancestry for each population, this is a principal component analysis, and the ancestral population are here. So you have the African population, the European on top, and then the Native Indians. So with what I tell you from Puerto Rico, could you tell me on this graph which is the population from Puerto Rico, the pink, the yellow, or the blue? Pink, exactly, because we say that Puerto Rico have a very strong, very weak Native Indian ancestry, but then a very strong variation on the European-African axis. So we see that each point is a different individual. We have those individuals that are very similar to African, those individuals that are very similar to European. Now, if you look at the blue are Mexicans from Mexico City, and you can see that they have a much stronger Native American component. And the yellows are from Buenos Aires and Argentina, where they have a much stronger European background. So when we were talking about the uniqueness of each Latin America population, you can see here that they are genetically very different, also in terms of their proportion of ancestry. So now if we compare the incidence of breast cancer, so coming back to our breast cancer, we see that compared to US whites, Hispanics in the US and Puerto Ricans have a much lower incidence of breast cancer. And we also know that African American, Hispanic, and Native American women living in the US, despite having a lower incidence of breast cancer, when they're diagnosed with a breast cancer, it's often diagnosed at a later stage, and it's often a more aggressive type of tumor that is going to be more difficult to treat. And some of this is related to access to care that might be a little bit more limited in some of those populations, but some of this is also due to genetic factors. So we wanted to look, and some studies have shown, especially in Mexican American, that the European ancestry was associated with a risk effect for breast cancer, and we wanted to see if that was the case in Puerto Rico. So we assessed the role of ancestry in predisposition of cancer in Puerto Rican women, and we also look at whether ancestry was a predictor of the tumor aggressiveness that we would see in the population here. So this is our population, so you can probably hopefully recognize the island, and you can see that the bigger the circle, the more samples are coming from a certain area. So most of our samples are from the Ponce area and from the San Juan area, but we do have a representation of all of the island. And so the study is now a little bit over 1,000 individuals, and we recruit breast cancer patients that are recruited at the moment of diagnosis and before getting any treatment, and women that had a negative mammography in the last 6 months. So those are our control group. And then for those samples, we get a whole bunch of things. So we get first informed consent, which is a necessary thing. We do have them to fill a very detailed epidemiological questionnaire about their lifestyle, habits, education level, all of those other factors that are important factors for cancer risk. We take a blood sample, but we also take a tumor sample and pathology data. And we do a whole bunch of study. And now I'm going to be talking about the admixture study, but we may touch upon other studies later. So we have that population of cases in control, and then we use those ancestry informative markers to assess what proportion of the genome of each individual is from which ancestry. And this is the results that we're getting. So what you see, you have, again, our ancestral population in the three different areas. And then we have the cases in control, which are represented as blue cross and yellow cross on this axis. And you can see, again, our variation is mostly on the European-African component with a little bit of a stronger European contribution, which is what we expected. But so when we look at this, what we see is that really the clouds for the cases and for the controls are completely overlapping. And if we do statistics on this, we find no significant difference in ancestry in our cases and in our control. And what we think is that what is seen in the Mexican American is the protective effect of the Native American ancestry, which we cannot really see in Puerto Rico because everyone has a very low ancestry from Native American, and everyone have a pretty constant ancestry from Native Americans. But so I think that's why we cannot see that risk effect of ancestry on breast cancer in this case. However, if we look at ancestry, in our cases, by grouping the cases by whether they had a tumor that was negative for hormonal receptors, so we can see that individuals that have PR, progesterone and estrogen negative tumors, or the triple negative tumors, have a lower European ancestry and a higher African ancestry. So that is telling us that that genetic ancestry is important in determining whether the tumor is going to be receptor negative or not. So, and then we didn't see any effect of ancestry on cancer type, tumor grade, or the presence or absence of lymph node invasion. And So what we can see is that in Puerto Rico, and this is a little bit different from the studies that were done in Mexican American, that we see that the global ancestry is not contributing to the risk of breast cancer, but it's contributing to the tumor hormonal receptor status in this population. And what this means is that if you have two individuals within the same population, and you're doing the follow-up of an individual that has a stronger African ancestry versus a stronger European ancestry, they're going to be at different risk of having tumor negative for those receptors. And so what we're doing now is that we're using that admixture data, and we're doing a panel of 2,000 markers that will tell us where in the genome those genes for determining the risk of hormonal receptor tumors are located. So we're going to be able to know exactly what genes or what changes in those genes are important for determining that effect of ancestry. And this is the paper for the Mexican study. So what we've seen so far is that while sharing a common language, and that puts aside Brazil, Hispanic and Latino populations show differences at the genetic level. And what that means for us researchers is that it's important that we do include Hispanic or Latino population in our studies, and that is true for clinical trial as well. And that can help us to reduce health disparities that are associated with different populations. So let me move to the next part. And so we've talked a little bit about the BRCA genes, and you're probably now expert by the end of the week about this. But what I wanted to touch upon in the previous talk was a nice introduction on those a little bit more problematic clinically, I would say, genetic variants that have smaller effect on risk and that act together with the environmental factor to determine a person's risk. And what we hope is that when we know them better and when we know enough of them, we could incorporate that data into the current prediction models, like the Gale model, to be able to give a better prediction of the risk for each woman. And so I'm going to move from the pie to a more, I'd say, more accurate representation of the contribution of genetics. And I like to show it as a continuous band because really we cannot draw a line between what is hereditary, familial, and sporadic, because there's always a little bit of genetic contribution or a little bit of environmental contribution. Even in BRCA carriers, most of them will develop breast cancer, but not all of them. So what is those extra factors that are making certain women, despite carrying the BRCA mutation, not developing a cancer? It might be environment, might be other modifier genes that we don't know yet. So really, when we think about breast cancer genetics, it's more a continuum between those cases with a strong genetic component, like the hereditary cases, and those cases with a very strong environment components. And at this end of the range, you can think, for example, of lung cancer in smokers. But still, some smokers will never develop a lung cancer, and some will. And why is this? Is because there's always a little bit of a genetic contribution as well. So we know of several risk factors that in the environment, including age, geographic location, lifestyle, reproductive event, mammographic density, and exogenous hormone. And there's more of those risk factors. Now, what's important is that how do our genes respond to environmental threats is that we have a mechanism that we call DNA repair. So anytime that you have an exogenous damage or an endogenous damage, for example, results of metabolism, you can have changes in your DNA. And the normal process would be that there's one of the DNA repair pathway that will find that damage and repair it. If the damage is repaired, the cell can return to cell division. If the damage fails to repair, then you would hope that the cell goes apoptosis. If it doesn't, then you have potentially the beginning of the tumor formation process. And we know that there's many different pathways that will repair many different types of damage. In fact, we think that there's more or less 150 genes in the genomes that are important for that DNA repair function, so that are important for those DNA repair pathway. Now, what's interesting is that most of the genes for hereditary cancers are DNA repair genes. So if you think of the BRCA genes, those are DNA repair genes. If you were thinking of the Lynch syndrome, those are also DNA repair genes. So many people have been thinking, well, if they're so important for hereditary cancer, could they also have a smaller impact in those more complex cancer? There's also, and the pathway specifically I'm going to be talking about today is a pathway that is called the nucleotide excision repair. So you know that Lynch syndrome would be a mismatch repair gene, and that's why you get that microsatellite instability in the tumor. Now, the BRCA genes would be more linked to the homologous recombination repair, and the nucleotide excision repair is a pathway that will detect any bulky lesion. So any change in your DNA that will kink the helix a little bit is going to be repaired by the NER pathway. And the NER pathway, when mutated, gives you the xeroderma pigmentosum phenotype. So those are individuals that are extremely sensitive to sun, but also have a 1,000-fold increase in risk of skin cancer and also a significant increase in risk of other cancers. So these are the genes that are involved in the NER pathway, and I'm not going to go into this in detail, but basically just like most DNA repair pathway, you have some proteins that are involved in recognizing the damage, and then they will bring other proteins that will remove the damaged segment of the DNA and then repair that specific damage. And so there's about 30 genes that are directly involved in the NER pathway. So since the DNA repair is so important for hereditary cancers, we wanted to see whether genetic variants, and I'm going to be talking about two specific types of genetic variants in DNA repair genes, could be involved in those non-hereditary cancers in Puerto Rico. So we're using the same population that I introduced before, but in this case, we're going to do something different with our sample. So we're going to be looking at a specific assays that measure how good is a cell to repair the DNA. So that's what we call the DNA repair capacity. And this is done by Dr. Mata in Ponce. And we're going to be genotyping SNPs and CNVs in lymphocytes of those patients. So what is the DNA repair capacity assays? It's what we call a whole cell reactivation assay. So what we do is that we take a plasmid that contains a gene for luciferase, right? So that's going to be kind of a reporter gene that we can track whether it's expressed or not in our lymphocytes. So we're going to take this plasmid and we're going to submit it to a UV light source. So that UV light is going to cause the type of damage that would be repaired by the NER pathway. Once that plasmid is damaged, then we're going to transfect it in our lymphocytes from patients and controls that have been cultured. So the idea is that once we transfect the plasmid, then the cells are going to use their endogenous repair pathway to repair the plasmid. Now, if the cell is good at repairing the plasmid, then the luciferase gene is going to be intact and it's going to be expressed. But if the cells are not so good at repairing it, then because the plasmid is damaged, then there's not going to be a lot of expression from the luciferase gene. So you can measure how good a cell is at repairing the DNA just by looking at how much luciferase activity you get in those cells. And I remind you that this is the lymphocyte of those patients. So we're not talking about the tumor cells. So we're really talking about the germline, basically the germline capacity of the cells to repair the DNA. Because if we were measuring in tumors, then we would be having a completely different or genetically modified population of cells. So by looking at lymphocytes, we're looking at what kind of DNA repair capacity people inherited in their germline genome, basically. So we look at two more types of genetic variants. So I've shown you the admixture data with the ancestry informative markers, but we also look at SNPs in those NER gene, and we look at another type of variant, which is called copy number variants. And I don't know if you heard of copy number variants before. Okay, no. So I'm pretty sure that throughout your studies, you've been told that humans, we are diploid organisms in all of our somatic cells. Well, recently, with new sequencing technologies, it was found that actually some part of our genome are duplicated or deleted. And so CNVs, and this was thought to be true for sick individuals. For example, if you were looking at people that have very severe disease, then you would expect those deletion or duplications. But it was found out that even in what we consider perfectly healthy individuals, we have those pieces of the genome that are present at various copy number. And it's thought that they could be contributing to disease just like in the same way as SNPs are contributing to disease. And if you think about it, if you have extra copy of a gene, then you might think that this gene might be expressed a little bit more. And some studies have shown that certain CNVs, when you have a duplication, for example, would be associated with higher expression from that particular gene. So sometimes by genotyping SNPs, you can also assess the impact of copy number variants, but not always. So we actually individually looked at those copy number variants and at those SNPs in the NER gene regions. And this is preliminary data. So we have started by looking at the SNPs that have a potential functional significance. So those would be SNPs that will change the coding of the protein. But we're also currently undergoing a large screening with hundreds of SNPs that will provide like a more complete coverage of each gene. So this is the characteristic of our study population. So we can see that our cases and control have a different age. And so this is not the ideal situation in statistics, but there's ways to correct for that when we do the analysis. And so on and so forth. So this is the information that we have for each woman. And we see that we have a difference in education level and I think contraceptive use in our cases and control. So for those factors, we're going to be correcting in the analysis. So this is Dr. Mehta's data just looking at DNA repair capacity in cases versus control. And what we see is that this is the distribution of the DNA repair capacity. And remember that the higher the value, the better the cells were at repairing the damage. So what you see is that in cases which are in red, the DNA repair values are much lower than in controls. So in fact, they're 56% lower when compared to control. So it's a pretty dramatic effect of cases between cases and control. Now, if we look at age, and I bring our attention to the age of the control, so this is again the DNA repair capacity, but in grouping cases and control in different age group. And for controls, DNA repair capacity is something that we lose with age. And so, there's many things that don't get better with time. Well, DRC is one of them. So sorry to give you one more preoccupation, which is age related. But in our cases, the effect is relatively, that age effect is not seen. But again, we see that our cases have a much lower DNA repair capacity than our control. So our hypothesis was that maybe part of it is due to the environment, but maybe some women are just born with weaker DNA repair capacity that makes them more at risk of developing a breast cancer. And so this is the first 8 SNPs that we typed, and again, we selected them because they were potentially functionally significant. And this is our most significant SNP. So it's a SNP in RAD23B, which is a gene that is important for recognition of the damage. So one of the first steps of the NER. And what you can see is that the distribution of the genotypes is drastically different between cases and controls. And for any of you that have experience working with complex traits, this is a very high odd ratio that we don't often see in complex traits. So basically, individuals that have the risk genotype, which is the TT, are 3 times more likely to get breast cancer than individuals that have the other, that are heterozygous or that have the protective allele, the CC. So that's a very strong odds ratio for a complex trait. Now, if this is a true association, what we should see is that this genotype should be associated with changes in DNA repair capacity as well. So what we see, and this is the first square that you have here, is that the risk genotype is significantly associated with a lower DNA repair capacity than the other 2 genotypes. And when we look at the association with DRC, we also see some significant, not as strong, but some significant effect on the gene XPC. And XPC is the gene that pairs with RAD23B for recognizing the damage. So because we're thinking of complex traits which have multiple genes involved, we thought, well, what happens if you carry a risk factor at both, at RAD23B and an XPC? So we did a model where we classified individuals by the number of risk genotype that they had at the 3 loci that were significant for DNA repair capacity. And what you see is that the more a person has, the more risk genotype a person has, the higher the odds ratio, or the higher chances that person have to have a breast cancer. And this is really an indication on how important it is when we study complex trait, and not to look at a subset of SNP, but I look at a global assessment of all the SNPs that are involved in your disease risk. And in this case, you can see that if you put all the 3 together, then you have a much stronger odds ratio. And what you can see if you look at the DRC for cases is that as you add those risk genotype, you can see that the DRC is progressively going down. So the people with the lowest DRC are the people with the most risk genotypes at those 3 loci, and the people with the highest DRC are the ones that have no risk genotype at those 3 loci. So it's really an example of what a complex trait is, where it's an accumulation of those smaller effect risk factors that actually gives you, in the end, a much stronger risk of breast cancer. Now, we also looked at CNV, and if ever you're interested in CNV, there's a database that is available online, and that puts together all of the results from a genome-wide search study where they have identified CNV. So you can type the name of your favorite gene, and you can see if anyone has ever seen a CNV in that gene region. So we did this for all of the NER gene, and these are those for which CNVs have been identified. So as you can see, CNVs are pretty common in the genome. And now again, we have looked at the association of our CNV genotype, which we classify as being less than 2 copy, which is the reference, which would be the case in a diploid genome, and then more than 2 copies. And we find out that DDB1 and RAD23A both have a significant association between the CNV copy number and the risk of breast cancer. But what's funny is that if you look at DDB1, having less than 2 copy actually gives you an odds ratio lower than 1. So that means that having less than 2 copy protects you from breast cancer, which doesn't make really sense, because we would think that DNA repair capacity is a good thing in protecting you from breast cancer, and you would expect that having more copies of a gene, of a mechanism that protects you against cancer, should be a positive factor. So we were skeptical about this, and we looked into it into a little bit more detail. And we went and we looked at whether these copy numbers were associated with DRC. So we would think that if they're really important for DNA repair capacity and cancer risk, we should see difference in DNA repair capacity, which was the case for RAD23A. But for DDB1, we didn't see any association between the number of copies and the DRC. So it really got us thinking as to what was going on. And we were thinking, because the association was pretty significant, it was a low p-value. So we went back, and this is the kind of data you can get from the database of genomic variants, and this shows basically your region of interest. And in our case, our gene is this one here at the end. And these are the CNVs that have been discovered, and basically the bigger the bar, the bigger the CNV, so you can see how many genes are included in a CNV. And this is the important point, because when we look at SNPs, we're looking at variants within a single gene, but when we look at CNV, we're looking at duplicated or deleted gene regions that are much larger and may contain more than 1 gene. So we thought, well, maybe the effect of DDB1 of the CNV in DDB1 at increasing cancer risk might actually be due to another gene in the same CNV that we just picked up by chance. And so if we looked at the genes that were included in those CNV regions, and we found a gene which is called VWCE. And this is a gene that has been shown to be associated with tumor progression and metastasis. And now all of a sudden, it makes sense that having more than 2 copies of this gene could increase your risk of breast cancer, because that would be a gene that would be more favoring the progression of cancer rather than protecting against. And we're now actually doing fine mapping of the region to confirm that hypothesis. But so far, it's the most likely candidate to be responsible for that association that we're seeing, which actually probably turns out not to be related to our DNA repair gene, but to another different gene on that part of the genome. And yeah, I think I'm gonna risk. So what we see, and so DNA repair capacity would be one of the pathways that is important for breast cancer. But we know and we expect that when we're going to want to think of all of the risk factors, the genetic risk factor for breast cancer, we expect that it's going to be probably a combination of DNA repair genes, but together with other genes, maybe the genes that talk between the DNA repair and the sense cycle, and so on and so forth. So this is part of the picture, but not a complete yet picture enough to be using that data in the clinic, unlike the BRCA data. But for sure, what we see is that for all age groups, the DNA capacity, repair capacity is lower in cases when compared to non-cancer controls. And we have found some variants, whether they're SNPs or CNVs, that were associated with changes in DNA repair capacity and, as a consequence, breast cancer risk. And that was true for Puerto Rican women, and it still has to be validated in other populations. Now, what's interesting is that when we combine the effect of multiple SNPs together, we can see incremental increases in risk. And we hope in the future, when we have finished screening all of the NER gene with all of the hundreds or SNPs that we have, together with all the CNV, that we can come up with a model that would integrate all of the DNA repair-related genetic variants, and that would give us a better prediction of risk. And so recently, there's been some talk about what is the best age for starting mammography screening, and this is all started because the U.S. Preventive Task Force suggested that maybe the mammography age should be increased to 50 because they were feeling that there were too many women that were at low risk and that were having too many mammographies from an early age. However, the American Cancer Society opposed. And so there's been a lot of talk as to what is the perfect age to start mammography screening. And from what you've heard with the BRCA work, and from knowing that basically every woman has a different risk of breast cancer, well, the answer is that maybe that age should be different for every person and should be based not only on family history, but also on environmental factors and on all of those genetic factors that we're going to identify are important for determining breast cancer risk. So we have to maybe target better our clinical management approach to different populations, but also within a population, not every individual is at the same risk. And so this is some people that have contributed to the work, including my students and technicians, especially Nelly, Alba, Julianne, and Hector. And then Dr. Mata has done a lot of work in recruiting that huge cohort that we're using. And then the people from UCSF, Elad Ziv, has been helping with the admixture. So you have any questions?